Our PiezoSleep systems detect and characterize rodent behaviors based on signals from non-invasive sensors. The system is designed to determine sleep and wake states of mice over short time increments based on pressure signals generated by contact with the cage floor. The critical step in product development is establishing a correlation between signal patterns and behaviors of interest. Classification algorithms in our system are based on intuitive signal features that have been tested against direct human observation and human scoring of EEG, EMG signals. The agreement between the algorithm predictions and direct assessment was around 90%, a little higher for mice and lower for rats. The performance can be compromised in situations where sensor signals are corrupted by high levels of noise or if the animal's signal is weakened. Therefore, it is important to ensure the cages are situated to limit exposure to excessive interference. This video describes how to use our signal quality analysis tool to inspect signal quality for a PiezoSleep system setup. The PiezoSleep system measures pressure signals from animal interactions with the cage using specialized sensors. Other contributions to these signals, such as vibrations from buildings, fans, or animals in nearby cages, are considered noise. A typical metric for assessing signal quality is the signal-to-noise ratio. This indicates signal power relative to the interfering noise signal and is often expressed in a logarithmic scale called decibels. An exact measurement of this value is very difficult because the signal power levels from the rodent vary with changes in activity. In addition, noise levels can change as well depending on building activities that create vibrations. The classification algorithms were designed to be robust to many of these changes in power values that would occur in a typical animal facility. The Signal Quality Analysis tool is a companion software that provides graphical views to check for signal quality and noise interference in files from PiezoSleep recordings. When assessing signal quality, it is best to find time intervals where the mouse activity is the lowest and building/lab activity is the highest. The most difficult separation for the sleep-wake classification is discriminating between sleep and low-level activity or rest. For the three-vigilance state algorithm, separating REM from NREM sleep is the most difficult. Both of these rely on extracting signal parameters related to subtle changes in respiration. If a breathing signal is consistently observed during periods of low activity, as shown here, that indicates good SNR. Note the rhythmic decreasing and increasing pressure from the thorax pressing on the cage floor, as well as some variations from small body movements or changes in respiration rates. If a strong interfering signal is present during respiration, as shown here, this can mask important changes in respiration and performance values may not be achieved. The Signal Quality Analysis tool provides a convenient interface to generate graphics and reports where the strength of the signal and noise can be observed over long periods of low activity. Details of how to use the tool, as well as other features not described here, can be found in the written manual. Here we open a sample file and selected one of the channels to review. At this viewing level, we cannot see the details needed to assess critical SNR, but we can observe expected patterns relative to the light and dark cycles. Let's zoom in to better fill the screen. This recording lasted for about 5 days. The vertical bars denote the light and dark onsets in the room. The x-axis is in hours, and the y-axis is in volts. This shows the pattern of rodent activity. High activity periods correspond to larger signal excursions because of the faster and more forceful body movements, and low activity periods correspond to smaller amplitude excursions because the animal has slower and more gentle movements or is still. Here, a nocturnal pattern is observed, where high activity patterns tend to occur more often in the dark phases, and low activity is more common in the light phases. The Signal Quality Analysis tool provides options for looking at 3-hour segments, where the starting hour of the selected segment can be entered here. Let's select hour 56 to observe the overall signal power, the time waveform, and the spectrogram. Graphics can be generated for single cages or exported to PDF files for all cages. For SNR assessment, there must be an animal occupying the cage of interest to generate a signal, and the recording must be long enough to contain extended periods of low activity. It is recommended that the recording be at least 24 hours so the cages are exposed to a full cycle of building activity and machine activation, such as from human foot traffic and ventilation systems. When testing a system SNR, all cages can be occupied, or some cages can be left empty to directly observe the interference patterns and power levels. The signal patterns observed in an empty cage are referred to as the noise floor. To generate graphics to help assess SNR, make sure the option to include the spectrograms and sensor snapshot are included. The best indicator of a sufficient SNR for the classifiers to work properly is being able to observe breathing signal patterns in the spectrogram over large blocks of inactivity. Now let's generate the graphics to analyze this segment. In looking at the time waveform, we can note a high activity burst for about a half hour starting around hour 56.5, then after hour 57, there are mostly periods of low activity broken up by periods of moderate activity, such as those near 57.5, 58.4, and 58.9. However, it is these periods of low activity that are most critical for assessing signal quality. Let's zoom in to observe better what is happening when the signal levels are low. Over this half-hour segment, bouts of stillness can be observed, broken up by short twitching, stirring, and posture changes. These flat-like regions at low levels illustrate the stillness patterns that have a high correlation with sleep, and when the animal is still, the only pressure on the cage floor is from breathing. This can be observed by zooming in further. Here it can be seen that the flat areas over the previous time scale consist of quasi-periodic variations that follow the respiration cycles of the animal. This is an example of a very good quality high SNR signal. If this pattern is observable throughout the recording, good classification accuracy can be expected as described in the manual. Zoom in further, and variations in each respiration cycle are observed. This pattern is used to separate rest from sleep as well as classify REM from NREM sleep. If this pattern is obscured by noise, then these classifications are more prone to error. However, it is very tedious to examine the time signal by zooming into these small intervals where the cycles from respiration can be observed. A less tedious way is to use the spectrogram plot. This plot indicates regions of activity and rest or sleep that are observed in larger ranges of time. This plot shows the spectral content as a function of time. Note that the x-axis is time and the y-axis is frequency. The spectrum is computed over short time intervals, and amplitudes are mapped into colors over the frequency axis. The color bar on the right is the mapping between signal power in decibels and colors. Here, yellow is relatively high and dark blue is low. In this way, the spectrogram shows signal energy over time and frequency. Signals created during active times tend to have high signal amplitude and fast transitions that tend to exhibit high energy at all frequencies. While during low activity, the quasi-period thorax motion from respiration only at the frequency associated with the respiration rate and integer multiples of that frequency. This is observed in the dark blue regions, where a horizontal higher energy band around 2 Hz is observed. There are also bands at 4 and 6 Hz, which are about 3 times the frequency of the lower band. This pattern seen in the spectrogram indicates good signal quality. If we zoom in on a smaller time region of the spectrogram, the change in respiration rate can be more clearly observed as the band moves up and down in frequency over this 8-minute segment. The location of the lowest band is referred to as the fundamental frequency, and the bands at integer multiples of this fundamental are referred to as the harmonics, as seen here by this lighter-colored, weaker band. The actual frequency for the respiration rate is that of the fundamental frequency. For rats, the respiration rates are typically between 1 and 2 Hz, as seen in the spectrogram here. Now we will look at the signals that are corrupted by noise sources that are typically found in cage setups and can reduce performance. In this case, we set up 6 cages on 2 shelves with 1 empty cage on each shelf. Vibrations of the platforms are often the source of noise, where the vibrations then resonate at certain frequencies based on the mechanical structures. Now let's look at the spectrograms of the cages on the top shelf. We can see the middle cage has much of its energy around 3 Hz, 4.5 Hz, and 7.5 Hz, which appears consistently over the whole time period. It does not vary in frequency or position like the breathing signal from a live animal. This is consistent with mechanical resonance or machine vibrations. From viewing the 3-hour interval spectrogram, we see a breathing signature in cage 1. However, we do not see bands for the noise signatures that are apparent in the empty cage. Now we will zoom in on the spectrogram for the first top shelf cages just after hour 56. We can see the spectral pattern for the breathing signal, and we can also see a constant 3 Hz signal, which is consistent with the noise signal in the empty cage. In general, noise from mechanical vibrations will typically be at a constant frequency, while biological variations like respiration will vary over time. Since we did not see these noise points on the bigger timescale view, it appears less than the actual breathing signal in the finer timescale view. This would be considered good signal quality, and the Village state detection algorithms should work as expected. If we zoom in on the time waveform from this region, a clear breathing pattern can be seen, with some impact of noise showing up as a small ripple during the negative excursion of the signal. For the features extracted from this waveform, this noise will have little to no impact on the classification. For the last cage on the shelf, we can see the noise resonance around 3 Hz and between 7 and 8 Hz. We also see that the spectral pattern of the breathing signal is not that strong. If we zoom in, we can confirm that they are at similar power levels. In the time signal, we see amplitude variations, and although a breathing signal is still observable, it appears corrupted, especially by the 3 Hz interference. This is considered reduced signal quality. And while the sleep and wake classification will have minor performance degradations, since we still see the breathing pattern in the spectrogram on the same level as the noise, the 3 Village state detectors will likely suffer more losses in accuracy since the algorithm is dependent on more subtle irregularities in breathing, which the noise may either mask or imitate. Based on the last example, we can see that one cage on the same shelf had very good signal quality, while the other cage just had fair signal quality. From the comparison figures, we can see the noise power is stronger in the last cage. Shelf position can make a difference in noise levels, since there may be different levels of stiffness in response to surrounding vibrations, proximity to a vibrating source, and exposure to wind currents from vents. Now let's look at the cages in the bottom shelf. Spectral patterns from the breathing signal are observed in both, along with the noise patterns. This is confirmed by zooming into the first cage. Here we can see an interval with stronger breathing signals and parts where it is weaker at the beginning and end. If we look at the time signal in the section where the spectrogram shows a weak breathing signal, we can see that the vibrations at the higher frequency mask much of the breathing signal. This will likely make it difficult to separate low-level activity accurately into rest and sleep. With a typical mouse, this can result in a loss of accuracy on the order of 5%. For the last cage, we see both strong breathing and noise, where the noise over all bands is slightly greater than the breathing signal. This will have a similar loss as the other bottom shelf cage. This video presented a few examples of identifying good signal quality from the spectrogram. The spectrogram allows us to observe large sections of time in low activity regions to observe the critical signal and noise patterns. It is recommended that every cage be tested with a live animal in the facility, where regular experiments will run for at least 24 hours. If some cages are left empty to observe noise patterns during an experiment, then another experiment should be run to have an animal in these cages. It is best to select 3-hour sections that have at least an hour of low activity and observe the spectrogram. If the 3-hour interval analysis is performed during an activity time for the building, this will provide an idea of signal quality during the worst case for SNR. If a breathing pattern is observed consistently stronger than observable noise, then the sleep-wake and REM-NREM wake classifiers should work as predicted. Thanks for watching.